In 2016, researchers built a Petri dish the size of a dining table. They filled it with black agar and divided it into bands. At the outer edges, there was no antibiotic. Move toward the center, and each band contained more. By the final band, the concentration was 1,000 times higher than the first dose that stopped the original bacteria from growing. Then they placed E. coli at both ends and turned on a camera. The bacteria spread as a white front. At the first antibiotic boundary, that front stopped. Almost everything there could go no farther. Then, at a few tiny points, white colonies appeared on the other side. Those colonies spread until they reached the next boundary. They stopped again. New pioneers appeared. The pattern repeated, step after step, until descendants of the original population were growing where their ancestors could not. The antibiotic had killed or halted nearly every bacterium it met, and the survivors had changed the future of the population. An antibiotic can work extremely well on susceptible bacteria, while leaving behind the rare cells that make the next infection harder to treat. Your body does not become resistant to an antibiotic. The bacteria do. Antibiotics interfere with bacterial machinery. Different drugs attack different jobs, building a cell wall, copying DNA, making proteins, or running some other process the bacterium needs to grow. For a susceptible cell, that interference can be fatal. But a bacterial population is not an army of identical little green dots. Every time cells copy DNA, mutations can appear. Some cells may also carry genes acquired from other bacteria. Most differences do nothing useful. Some are costly. A very small number may matter when a particular antibiotic arrives. The bacterium does not inspect the prescription, stroke its chin, and design an appropriate mutation. Evolution has no meeting. Attendance would be difficult. Variation comes first. The antibiotic then changes which variants leave descendants. Imagine 1 million susceptible cells and one cell carrying a useful resistance trait. Before treatment, that trait may offer no advantage. It may even slow its owner down. Once the antibiotic removes the susceptible competition, the same trait becomes an excellent piece of biological real estate. The resistant cell divides. Its descendants inherit the trait. What looked like one exceptional bacterium becomes a population. The megaplate made that selection visible, but it also showed something less tidy. The most resistant mutant did not always lead the advance. Some highly resistant lineages became trapped behind neighboring bacteria that had reached the frontier first. Space, timing, competition, and ancestry shaped which branch moved forward. The plate showed evolution spreading through a crowded landscape, one available route at a time, with no guarantee that the most resistant lineage would be the one in front. And bacteria have more than one route around an antibiotic. Some reduce the drug's access by changing the channels through their outer membrane. Some use molecular pumps to throw the drug back out. Some produce enzymes that cut or chemically alter it. Others change the molecule the antibiotic is supposed to bind or reroute the process the drug was meant to block. Resistance is usually specific. A bacterium may resist one drug and remain vulnerable to another. A resistance mechanism can also carry a biological cost when the drug is absent, but bacteria have a second inheritance system that makes the problem move faster. Parents pass DNA to descendants. Bacteria can also exchange mobile pieces of DNA between cells. One common vehicle is a plasmid, a small loop of DNA that can carry resistance genes. During a process called conjugation, two bacteria make direct contact, and one can pass a copy of a plasmid to the other. Other routes can move DNA released into the environment or carried by viruses that infect bacteria. It is inheritance with a suspiciously relaxed attitude toward the family tree. A useful resistance gene does not always have to wait for one successful lineage to reproduce. It can move into another bacterium, sometimes even across species, and travel with other resistance genes on the same mobile element. A resistance trait can therefore spread twice, down a lineage as bacteria divide, and sideways as mobile DNA moves between cells. Antibiotic exposure can select resistant organisms within a person, animal, wastewater stream, farm, or hospital. Those organisms and their genes can then spread between people, animals, food, healthcare settings, and the environment. The megaplate is not a tiny hospital. Its antibiotic bands were deliberately built to study evolution in space. A real infection includes an immune system, tissue, changing drug concentrations, competing microbes, and clinical decisions. But the plate reveals the central mechanism with unusual honesty. Killing most of a population is not the same as making evolution impossible. The phrase "antibiotics stop working" gives the chemistry too much blame. The antibiotic molecule has not forgotten its chemistry. Susceptible bacteria may still be killed by it. The population causing the infection now contains enough bacteria that can restrict, remove, destroy, evade, or bypass the drug. Doctors may need a different antibiotic, a combination, a higher level of diagnostic information, or another clinical strategy. Some resistant infections leave very few effective options. The plate lets us watch one bacterial population change. Surveillance asks how often medicine now encounters populations that have already changed. In the World Health Organization's 2025 surveillance report, roughly 1 in 6 laboratory-confirmed bacterial infections causing common infections worldwide in 2023 was resistant to antibiotic treatment. The dataset covered more than 23 million confirmed cases reported through surveillance systems, with large differences between regions and important gaps in reporting. For the pathogen-antibiotic combinations that WHO could track from 2018 to 2023, resistance increased in more than 40% of them. Those numbers do not mean that 1 in 6 people or 1 in 6 bacteria everywhere is resistant. They describe specific confirmed infections, pathogens, drugs, countries, and surveillance data. The boundaries matter. Antibiotics also support much more than the treatment of a sore throat or urinary infection. Surgery, organ transplantation, cancer therapy, neonatal care, and treatment that suppresses the immune system all become more dangerous when bacterial infections are harder to control. So how do we stop evolution? We do not. We reduce the opportunities it can exploit, and we slow the spread of what has already evolved. That means preventing infections through sanitation, vaccination, safe healthcare, and infection control. It means using diagnostics to distinguish bacterial infections from illnesses an antibiotic cannot treat. It means choosing an appropriate drug, dose, route, and duration for the actual infection, then following current clinical guidance, rather than improvising with leftovers. It also means surveillance, access to quality medicines, better stewardship in human and animal health, and continued development of antibiotics and other ways to prevent, diagnose, and treat infection. Any antibiotic use creates some selection pressure. Necessary treatment is not a moral failure, and withholding effective antibiotics from people who need them is not stewardship. The practical goal is to avoid exposure that brings no clinical benefit, while making effective treatment available when it does. Return to the giant black plate. The white frontier records every place where one bacterial lineage stopped and another found a route through. There is no moment when the entire species suddenly understands the antibiotic. There are deaths, divisions, mutations, transfers, bottlenecks, and the occasional cell whose descendants inherit the open ground. The medicine did not train an enemy on purpose. It changed the contest. Mutations and transferred genes supplied variation. Selection kept the variants that could continue. Antibiotics remain among the most useful tools medicine has ever built. Preserving them means reducing useless exposure, stopping resistant bacteria from spreading, and keeping effective treatment available for the people who need it. Stay safe.